Hello, my name is Gretchen Johnson, and I am a second year PhD student in Dr. Dorota Stankowska's lab at the University of North Texas Health Science Center in the Department of Pharmacology and Neuroscience under the North Texas Eye Research Institute. And today I'll be talking about some of the work that I have done to start investigating the mechanisms of peptain-mediated neuroprotection in retinal ganglion cells. Glaucoma is a group of multifactorial neurodegenerative diseases of the eye, and it is predicted that by 2040, around 112 million people will be affected. In most cases of glaucoma, loss of sight is gradual and accompanied by an increase of intraocular pressure, also called IOP. The buildup of pressure pushes on the back of the eye, specifically the optic nerve head, and the axons become damaged, leading to the death of their cell bodies, the retinal ganglion cells. So elevated IOP is a major risk factor in glaucoma that is accompanied by numerous cellular events and metabolic events that produce damages to RGCs, including mitochondrial dysfunction, apoptosis, and excitotoxicity. Current therapeutics target the change in IOP either through increased outflow or decreased inflow, but still only slow the progression of RGC degeneration. Thus, there is a need for adjunct therapies that protect the cells from other pathological insults. One avenue neuroprotection research has taken is through a protein called alpha-B crystalline, or CRIAB, a small ATP-independent heat shock protein that is found in the lens with high levels of antioxidant activity. Over the years, research has uncovered many other roles that CRIAB is able to play, including chaperoning, anti-apoptotic, and anti-inflammatory in the context of the eye. Additionally, its expression was found to be decreased in retina of IOP-elevated brown Norway rats. Our lab took an interest in this protein and decided to focus on a core fragment we called peptain with beneficial effects in the context of glaucoma. For this project, we conjugated cell-permeable peptide, called CPP, to peptain and named it P1-CPP. The purpose of the following was to determine mechanisms underlying neuroprotective effects of P1-CPP in retinal ganglion cells, RGCs, an ex vivo human retinal explant model, and in vivo in a rodent model of glaucoma. We hypothesized that P1-CPP will preserve retinal ganglion cells in both models. RNA sequencing results obtained from adult rat primary RGCs following IOP elevation will uncover some of the mechanisms by which P1-CPP mediates neuroprotection. First, I wanted to discuss some of the exciting work that we have been able to do through our World Body program, where we obtain eyes less than 12 hours post-mortem and isolated the retina by cutting at the optic nerve head, severing the axons to create an axotomy model. We then took 4mm biopsy punches from around the macula and placed RGC side up onto a transwell membrane submerged in media for 7 days ex vivo. To start, RGCs were stained with marker RBPMS, and cell counts were performed on punches fixed at day 0 when we isolated, as well as those incubated for 7 days, treated with either vehicle or P1-CPP at a concentration of 12.5 micrograms per milliliter. In a separate set of experiments, a total of 9 brown Norway rats were used, with 3 in the naive group acting as a healthy control, 3 in the IOP elevated group treated with vehicle acting as a disease control, and 3 in the IOP elevated group treated with P1-CPP. The IOP elevated groups underwent Morrison surgery, which is the injection of hypersaline solution to the episcleral vein, to elevate IOP, and then had weekly intravitreal injections until euthanasia on day 14. Next, we performed a unique procedure of isolating RGCs from retina using a double immunopanning method on adult cells, which are harder to obtain high enough quality data for sequencing. Thus, following IOP elevation, just these cells of interest were isolated and sequenced for total RNA per sample. Then this data was put through bioinformatics software to pull out differential gene expression. And then these results were then put through QIAGEN's Ingenuity Pathway Analysis software, or IPA, for functional assessment. After obtaining IPA results, confirmation began by performing quantitative PCR on RNA from sequenced samples, as well as performing immunocytochemistry on rat pup primary RGCs deprived of neurotrophic factors for 48 hours, co-treated with either vehicle or P1-CPP to mimic glaucomatous insult. Starting with the human explant data, as shown in figure 1b, the 0 days ex vivo group showed distinct staining of RGCs, and 7 days ex vivo of neurotrophic factor deprivation showed a significant decrease in total RGCs by almost 50%. In the P1-CPP treatment, at 7 days significantly prevented RGC loss by 35% compared to the vehicle. For the differential gene expression data from the in vivo rodent model of glaucoma, the first comparison made is between our healthy and disease groups, where there were a total of about 10,000 genes significantly differentially expressed. These differentially expressed genes I will refer to as DEGs. This is plotted in panel A, showing the fold change where positive FC is increased expression and negative FC is decreased expression. In the second comparison, shown in panel C, between P1CPP treated groups and vehicle treated groups, there are about 6,000 significantly regulated DEGs with the relative spread of increase decrease shown here. When assessing what these genes were, we found that 6,275 of the genes altered in the IOP vehicle compared to the healthy were also being changed when the IOP vehicle expression was compared to the P1CPP expression, indicating a change in pathology mediated by P1CPP. When the data was run through IPA, which uses databases of published literature to pull information about each gene and what pathway it may be connected to, the graphical summary returned is shown in figure 3, with potential mechanisms being represented by a single node. When looking further, we found several significant pathways, but decided to focus on these shown in figure 4A, specifically synaptogenesis signaling and Krebs signaling in neurons, which were the two most significant pathways. In heat maps, B and C show the genes that were identified as members of these pathways, and the fold change is displayed by color. Overall, there is a good indication that P1CPP reversed IOP-mediated pathological changes. First, to confirm results from RNA-seq, we focused on Krebs signaling pathway, an important player in cellular phenomena such as synaptic plasticity, cell proliferation, memory and learning, and neuroprotection. In panel 5A, the quantitative PCR results are shown where we confirmed an increase of CREB1 expression at the RNA level from the sequence samples. Additionally, we analyzed the protein data by immunocytochemistry of RGCs from 48 hours of neurotrophic factor deprivation, shown in panels 5B through D. P1CPP protected RGCs from death, as evident from 5B, as well as caused significant increase in CREB and phosphorylated CREB in panel 5C, to show an increase in activation as well as general expression. Panels D1 and D2 are representative immunocytochemistry pictures at 40x and 10x magnification. So in conclusion, P1CPP was neuroprotective against neurotrophic factor deprivation-mediated RGC loss in human retinal explants. Mechanisms of action of P1CPP in a rodent model of glaucoma includes the activation of the pro-survival Krebs signaling pathway and synaptogenesis to prevent cell death and vision loss. There is ongoing work into confirming the rest of the members of this pathway and others. My next step for confirmation is to first repeat the Morrison model to collect tissue, which can be used for immunohistochemistry or further protein assessment, various fluorometric assays such as calcium flux and caspase activity, and qPCR on isolated RGCs. Thank you so much for your attention. I would like to acknowledge my lab members as well as everyone at the North Texas Eye Research Institute, my funding from the Neurobiology of Aging and Alzheimer's Disease T32 grant, and the pilot grant awarded to my PI, Dr. Dorota Sankowska from UNTHSC. And finally, my references are linked here through this QR code in the bottom.